down. Simultaneously, we have an expansion of tidal volume because of an increase in end inspiratory lung volume. So during exercise, in healthy individuals, tidal volume expands by increasing the end inspiratory lung volume into the available inspiratory reserve volume, and by decreasing the end expiratory lung volume into the available expiratory reserve volume. So this is important because this pattern or control of operating lung volumes, as you'll see in the next slide, allows humans to expand their tidal volume by staying on the linear portion of the respiratory system sigmoid pressure volume curve. Okay, so just as a reminder, remember, tidal volume expands up to about 60% of vital capacity and then plateaus. And we said that this was probably mechanically advantageous because if we breathed 100% of our vital breath, we'd be breathing on every expiration down to residual volume where there's a lot of resistive loading, and on every inspiration towards TLC where there's a lot of elastic loading. So we'd be putting a pretty big burden on the respiratory muscles to overcome these resistive and elastic loads. So if we come back to the pressure volume curve, is that at rest we have a tidal volume that looks like this. And then tidal volume during exercise, as I showed you previously, will actually expand by decreasing from rest the end expiratory lung volume and increasing from rest the end inspiratory lung volume. These end inspiratory and end expiratory lung volumes will encroach on the expiratory and inspiratory reserve volumes, but all the while tidal volume will expand along the linear portion of this curve where the respiratory system is compliant, where the work of breathing or the amount of pressure that the respiratory muscles need to generate to support a given tidal volume is relatively modest. We're not forcing the body to generate disproportionately large efforts to achieve a particular volume change. And so this behavior, or the regulation of operating volumes, the control of end expiratory and end inspiratory lung volumes are very important because remember, one of the challenges of the respiratory system was to support 20 to 30-fold increases in ventilation while minimizing the work and the oxygen cost of breathing. This is how, if you can support 20 to 30 time increases in ventilation while minimizing the magnitude of increase in the work and the oxygen cost of breathing. So let's say, for example, that you had to achieve the same pink tidal volume expansion, but you didn't have the exercise-induced decrease in end expiratory lung volume. End inspiratory lung volume would be positioned where? Higher or lower to TLC? This pink curve would shift up, right? So it would be higher by the amount to which the normal response would obviously cause a deflation. In other words, if you did not have the reduction in end expiratory lung volume, but you had the same tidal volume expansion, this end expiratory lung volume would actually be positioned right up here, which is not advantageous. So again, to anchor that right now, everybody take a breath in till you're about 95% full, and then try to breathe up there for 10 seconds. So the behavior of operating volumes is preventing you during exercise from breathing there. So by decreasing your end expiratory lung volume and increasing your end inspiratory lung volume in the way that it occurs, it keeps you on that linear portion of the curve, which satisfies one of the requirements or the challenges that's faced by the respiratory system, which is to support very high levels of ventilation while minimizing the work and the oxygen cost of breathing. You'll see in exercise patho, for those of you that take it with me, is that this goes completely wrong in restrictive and obstructive disorders because of various pathophysiologies of the lung and chest. And so people are forced to breathe up on that alinear portion of the curve very, very quickly. They become very uncomfortable very quickly. Okay? So the control of dynamic operating lung volume, specifically end inspiratory and end expiratory lung volume, during exercise allows tidal volume to expand within the compliant or linear portion of the respiratory system's S-bend curve, where both the resistive and the elastic work of breathing is minimized. Okay? And it's important for you to have an appreciation that the elastic work of breathing is on inspiration. Okay? End inspiratory lung volume encroaching on total lung capacity, It's the inspiratory muscles that have to work against the elastic recoil and the stiff chest wall. So if you were asked a question on an exam, is it an expiratory elastic work or an inspiratory resistant work associated with breathing at a high lung volume? It's inspiratory. The resistant work is occurring at low lung volumes. At high lung volumes, it's largely an inspiratory elastic work. Okay? So that is a little bit descriptive, not particularly mechanistic, but shows like, so it shows you the pattern of breathing, the operating volumes, and just basically the pattern of response during exercise in a healthy, normal individual. The question becomes, what are all the mechanisms trying to control these factors? What's responsible for this pretty precise control of ventilation and matching the metabolism? It's complicated. There are a lot of factors, and they're very redundant, and the system is incredibly plastic. Okay, so it's basically, if I chop off, if somehow I were to eliminate one nerve pathway, it seems like another one upregulates. It doesn't seem like any one of these pathways are obligatory to, in other words, responsible for 100% of the ventilatory response to exercise. Because I'll show you some evidence that in their absence or presence or whatever it is that you still ventilate. You just might hypoventilate a little bit, but you don't eliminate ventilation altogether. There's a lot of redundancy in the system. The point is that you have a couple of factors and a couple of pathways that can contribute both to the feed forward and feedback control of ventilation. So we have central command. So basically we have these neural feed forward mechanisms that originate in higher brain centers that can control ventilation independent of the periphery of metabolism. So for example, you can hold your breath right now. That's central command. You can override your respiratory control pattern generator and control cortically through higher brain centers your own ventilation. You can also hyperventilate. So that is a form of central command. We're driving ventilation independent of feedback related and say metabolically related influences. Okay? So we do have this central command mechanism that this is a neural feed forward control originating higher brain centers like the motor cortex and the hypothalamus. This will send information through the phrenic and the vagus nerves to activate the lungs and the respiratory muscles. And we also have a series of afferent responses or feedback responses that can contribute to the control of ventilation. So we have chemical and chemoreceptors or metaboreceptors and mechanoreceptors. So type 3 and 4 afferents that exist in the muscles that can respond to hydrogen ion, different ions, movement and vascular distension, and temperature. So we have all of these kind of inputs that at the muscle, which is really the site of metabolic demand during exercise, can signal to the respiratory controller, increase ventilation. We're working. We need more gas exchange. We need more nutrients delivered, more byproducts removed. The lungs are an important component of that, so increase ventilation. We also have receptors in the lungs themselves, so stretch and flow receptors that can signal levels of appropriateness about ventilation. I think I talked about the pulmonary stretch receptor and the Herring-Brewer reflex, where lung stretch actually results in an inhibitory signal to turn off ventilation. Right, so the lung stretch sends an inhibitory signal to transition between inspiration and expiration. And so again, we have these afferent feedback signals that are originating from the lung. We can also have respiratory muscle metaboreceptors. So if the respiratory muscles become basically fatigued and they develop metabolites because they're overworked, that also can signal information to the respiratory controller to modify ventilation. We have peripheral or carotid chemoreceptors which respond to a host of stimuli. So again, hydrogen ion, various ions, the arterial CO2 and other humoral stimuli. And then we also have the medullary chemoreceptors that are largely responsible for responding to changes in cerebral spinal fluid hydrogen ion levels. So we have all of these candidates that can contribute to the control of ventilation during exercise. That's all this slide is meant. There's many different factors that can contribute to the control of ventilation during exercise. So what does the ventilatory response to exercise look like? So if we went from resting right now and automatically I just said go from rest and then start exercising. So start doing a constant load exercise test. Okay? The ventilatory response looks something like this. Ventilation rest, and then a step increase in exercise intensity, in this case arbitrarily of 100 watts. So from rest, at the onset of exercise, you have an immediate, almost a within-breath increase in ventilation. It's remarkably fast, which means it's probably not a feedback-mediated phenomenon, because feedback-mediated phenomena require an error. More likely some type of fast feed-forward or central command-related phenomenon. I'll show you some evidence of that, because it's the only thing that's really fast enough in our control system to result in a within-breath increase in ventilation on that magnitude. Just consider it time. You need to build up an arrow signal for chemoreceptors and feedback-related systems to activate. So within a breath at the onset of exercise is probably not fast enough for that to happen. And then in the transition from exercise, you get this phase 2 response that occurs where you get this progressive or exponential rise in ventilation that typically lasts 30 to 90 seconds. This looks a lot like the blood flow profile, right? Immediate rapid adaptation, then you get this kind of progressive adaptation to achieve a new steady state. It also looks like the VO2 adaptation, right? VO2 rises and then basically progressively changes. And we said that we had the time course of the VO2 onset kinetics. And anyways, the idea is that there's always these adjustment periods where we're trying to achieve a new steady state. And then we have the phase 3 response where ventilation is now proportionate to the rate of carbon dioxide production. Right? So the idea is that we have this 3-phase response. The black box of kind of respiratory physiology is what explains this immediate rapid increase in ventilation, because it occurs so fast and it's very, very hard to study. And you can do stuff in animals, but doesn't necessarily translate into humans. So I'll show you some animal and human studies that, again, provide evidence or for a role of these various efferent and afferent pathways in the control of ventilation during exercise. Before we do that, I just kind of want to show you what the ventilatory and gas exchange response looks like in the rest of exercise transition going from light, moderate, and heavy intensity exercise. So if you go in light intensity exercise, as I showed you previously, ventilation will rise immediately. The rapid increase in ventilation, as you can imagine, metabolism hasn't really changed all that much. So what happens is that you actually get some changes in PO2 and PCO2 that occur, and the exercise intensity is pretty modest, so you're not getting any disturbances in blood lactate or hydrogen ion. During moderate intensity exercise, you're getting a larger phase 1 response. Now, ventilation needs to try to support a higher metabolic demand, so the magnitude of the response is going to be elevated. During moderate intensity exercise, you're really not getting much in the way of gas exchange disturbances nor metabolic disturbances in terms of lactic acid and hydrogen ion production. During heavy intensity exercise, what happens is that you have a much more dramatic phase 1 response, and actually you don't achieve a steady state. Because now we're exercising at an intensity above the ventilatory threshold, where now ventilation needs to compensate for the metabolic acidosis, and so we have this situation where ventilation will be disproportionately greater than the rate of carbon dioxide production, as I showed you a few slides ago. And under these circumstances, you can see that the alveolar PO2 will rise. An individual will blow off carbon dioxide levels pretty quickly as a means of trying to compensate for this metabolic acidosis that occurs quite quickly in the transition from rest to heavy intensity exercise. So let's say that you did like a Wingate test, for example, would be kind of like the extreme example of this. You go from nothing to 30 seconds of an all-out response. You would get this very dramatic increase in ventilation with no achievement of a steady state because you're exercising at near maximum intensity. So your body's not going to be in a steady state condition. Okay? An important component here is that, remarkably, this phase 1 component of ventilation, this immediate increase, is intensity dependent. So we have something that is able to increase ventilation in an intensity dependent manner and do it very, very quickly. So it's kind of interesting, concentric command, like, is there, proprioceptive receptors that at the onset of exercise is like, oh, that's a pretty big load, send information to the brain to drive ventilation quite aggressively versus, oh, that's not hard. Not much disturbance or perturbation in my mechanoreceptors, don't have to drive ventilation all that hard. So there is something that seems to be intensity dependent here. So let's consider some of the factors that could be responsible for this immediate increase in ventilation, so this phase 1 response. Not particularly concerned right now with the phase 2 and the phase 3 responses. So what are some possible candidates from that big flow diagram that I showed you previously? Well, one is certainly central command. So we could have feed-forward control of both locomotor and respiratory muscles, and this is really descendant efferent information originating in higher brain centers, the cerebral and motor cortices, and the hypothalamus. So presumably this would be very, very quick, right? Because in some ways you have some voluntary control over it. Breathe faster now. Like, do that immediately, right? So it can happen very, very quick. So central command could certainly be one of those factors. You could have neural input from type 3 mechanoreceptors or sensory afferents. So these are basically sensory nerves located in the respiratory and locomotor muscles, as well as the joints, tendons, and blood vessels. So in terms of speed and rate of change, as soon as your muscle contracts and you get some proprioceptive stimuli, while it's not feed, it is an afferent signal, but it's kind of clustered into being a fast neural feed-forward response, because it's not responding to an error per se, right? So you get this mechanoreceptive stimulation. Say, for example, with the release of, you get the muscle pump, you get this immediate increase in blood flow that causes vascular distension. The mechanoreceptors in the blood vessels send information about, okay, my muscle's doing something. Increased ventilation. So you can have this very fast kind of neural feedback response that actually operates within the feed-forward domain of respiratory control. Does that make sense? Again, it's just the extent to which it is an afferent nerve, but it fires so quickly that it's kind of considered part of the feed-forward system. And I think there was some confusion about that. Somebody had asked Yasmin, I don't think I answered that email, but somebody asked Yasmin about there was the mechanoreceptor, it was talked about as a feedback mechanism, but then somewhere else it was talked about a feed-forward. It's technically feedback because it's an afferent nerve pathway, but it's so fast that we cluster it in to kind of the feed-forward response. And it's a similar mechanism here. Then we have neural input from type 4 metaboreceptors. So these are chemical sensitive nerves that exist in the respiratory and locomotor muscles that would respond to metabolic disturbances. So at the onset of exercise within a breath, does the muscle become acidotic? You know, no, it's too fast. So these chemical sensitive receptors are probably not necessarily responsible for that immediate increase in ventilation. So the question is, have the muscles had enough time to increase the metabolites, therefore stimulating these sensory afferents? Probably not that quickly. And then we also have the central and peripheral chemoreceptors, so in the brain and at the carotid bodies. Have the muscles had enough time to increase the metabolite concentration in arterial blood within a breath? No, because remember, even during light and moderate intensity exercise where there's no acid-base disturbance, ventilation increases in an intensity-dependent manner at the onset of exercise. Does the blood circulate from the muscle to the lungs, through the heart, the central and peripheral chemoreceptors in less than 5 seconds? Transit time of a red blood cell through the body is about 6 seconds. So, still, not necessarily within the time window that we're looking at. And does arterial blood and cerebral spinal fluid hydrogen ion levels increase as a stimulus for these chemoreceptors during mild and moderate intensity exercise when there is a step increase in ventilation? No. So there really doesn't seem to be a time-dependent or stimuli-dependent activation of these receptors. At least, again, in that immediate increase in ventilation. So, we will end with, actually, let's end with this. So these are what we're going to do now on Tuesday, and then moving into Thursday. Well, we'll finish this on Tuesday. And I talk really slow. For some reason, I can't get through the slides that I present, but it's just kind of what happens. So those that will have me just know that if I put up 35 slides, I finish 25, and it's just always the way it goes. Point is that we'll finish this next class, which going actually through experimental evidence. This actually study being pretty cool, where they have basically a brain dead animal that they stimulate and it moves, and then they paralyze it, and then it doesn't move, and then they measure all these nerve recordings. It's pretty cool. Anyways, this is like, I wouldn't do it myself, but this is the not as a subject, as an experimenter. But the idea is that this is kind of like the major study that described the role for central command and control of ventilation. So what we'll do in the next couple of lectures is we'll actually go through experimental evidence for and against these different feed-forward and feedback pathways contributing to the control of ventilation during exercise. And then we'll move into a series of lectures about how, or a series of slides about how the respiratory system actually can be a factor contributing to exercise performance limitation. And then you have an exam grade. Alright? So, do well with the cold, and I will see you on Tuesday.